my name is Jordan Garo. I am presenting my paper on neuroeconomic tools. This is for MBA 639. Neuroeconomics is a multidisciplinary study of neuroscience, psychology, and economics. It is only a few decades old, and it is trying to determine the neurobiological basis for decision making. There are multiple tools in order to study the neurobiological basis of decision making. The first, and one of the older ones, is the EEG, or electroencephalogram. It has a high temporal resolution, which means that it records very quickly. So if there is a sudden and abrupt change in electrical activity, the EEG will pick it up. However, the EEG only picks up action potentials across the scalp and very surface of the cortex. It doesn't actually go into any depth into the cortex for any kind of resolution. But it is low cost, and so it is an important tool for researchers examining specific questions related to speed of response. The next tool is similar to EEG. It is non-invasive. It's just used over the scalp, and it's called the MEG, or magnetic electroencephalogram. It has a high temporal resolution, just like the EEG, but also low spatial resolution. But this does go deeper into the cortex in order to determine where the activity is occurring. But it is high cost. It involves this cyclotron, but it is good for fast responses as well as the EEG is. But this just has much better resolution. Another tool of the neuroscientist toolkit is the PET, the positron emission telemetry. It is used to you put a tracer into the bloodstream of a patient, and it is a radioactive tracer. It's attached to a molecule such as glucose, and as glucose is the fuel of neurons, when neurons are firing rapidly and engaged in heightened activity, they are absorbing this glucose, and it is shown on the scanner through regions that are expressing high activity. It is low temporal resolution, so it is not used to find quick responses. It is more of a snapshot rather than a real-time look at what the brain is doing. But it has very high spatial resolution, down to just a few millimeters. But it also is very high cost. But it's very good for determining location accuracy. So you'll take two pictures, one that is more of a control picture of nothing, and then one that is a picture of the activity that you're looking for and localizing, and then you lay these on top, and whatever the difference is, that's the region that you're trying to pinpoint for whatever question you're trying to answer. The fMRI is the most used tool of neuroscientists. It has good temporal resolution, so it operates quickly, but not instantaneously. It has a slight delay of a few seconds, but it has very high spatial resolution. It is also very high cost, and it is excellent for location accuracy. So it can look in 3 dimensions across cross sections of the brain to determine where exactly the activity is occurring. And how this machine works is it creates this immense magnetic field that aligns all the protons within hydrogen atoms that are located within the water molecules in your body. So it aligns all of the hydrogen protons into alignment with the magnetic field that is generated by this machine. But the machine also sends out small radio frequencies in order to bump those protons out of alignment. And when it bumps them out of alignment and then goes back into alignment, it emits this energy that is able to be picked up by the fMRI machine. And so what it basically does is it can find water molecules within your body. And those water molecules are located within your bloodstream. And the bloodstream is a good indicator of where activity is occurring in the brain, because if there is a region of the brain that is heightened in activity, it'll also have more blood flow. So basically what this machine does is it localizes increased blood flow to regions of the brain to determine where the processing of whatever question you're trying to answer is occurring. This is by far the most widely used tool of all of neuroeconomic and neuroscience research. There are other tools, also non-invasive. This one is relatively cheap compared to some of these other ones. This is called transcranial magnetic stimulation. It doesn't actually, it's not actually a tool used for imaging. It's more of a tool used to simulate brain lesions. So what you use this tool for is to essentially shut off regions of the brain and basically by the behavior of the patient or their personal responses to what is happening to them, you can determine what that region of the brain is used for. So you can turn off someone's ability to speak if this is located over the BRCA region of the brain, or if it's over the occipital lobe, you can make them temporarily blind. And this is important that it is just a temporary thing, that it only lasts for a few minutes, but it's a really interesting kind of tool. And this one, optogenetics, is a brand new method that is not being used in humans. It's only being used in mice, but it's very interesting. What researchers have been able to do is they've injected a photosensitive gene from a jellyfish into specific neurons within regions of the brain that they're trying to target. They've identified these genes that are promoting the development of specific neurons that they're trying to research. And so when they inject the gene, these neurons grow with this photosensitivity. And how they use that photosensitivity is they run a thin fiber of fiber optic, as you can see in this picture of the mouse, directly through the cranium. And so they can turn it off like a light switch. And as the light hits those photosensitive neurons, they fire. So you can basically turn on someone's fear response, or someone's anger response, or any number of things at the flick of a switch. Again, this is only used in mice studies and has not been used in human trials, and will likely not be used. But the important thing of this is that you're getting beyond just the structure of the brain. You're getting to the underlying code that is building the structure of the brain. In order to pinpoint where the specific neurons are going to be, you have to understand which gene is in the development of that neuron. And if you understand the genetic basis of these regions, you can really do a lot to play with how these, to augment these regions of the brain to do any number of things. And so that gets into the new frontier of possible gene editing or genetic engineering with CRISPR-Cas9, which is a uh, really interesting genetic tool that can basically swap in and out DNA anywhere in the whole of the DNA with pinpoint accuracy. This permanently changes the DNA of the subject, and it can change it in whatever way the researcher deems fit. Obviously, this is wrought with very substantial ethical issues, but there is a potential for something like this to take place. And what could, if we're talking about augmenting the brain, what kind of behavioral changes, and especially in decision making, what kind of changes are possible? Can we make somebody more rational? Or can we make them more compliant? Can we make them more docile? Like there are a lot of potential ethical issues involved in any kind of research with gene editing. But when it comes to changing somebody's fundamental personality and their free will, essentially, um, it, you're hitting walls that probably shouldn't be broken. So that is my presentation, and if you have any questions, feel free to ask.